Hi everyone, Brenda Gustin here, and we're going to continue our discussion about carbohydrate catabolism. So remember we talked about aerobic cellular respiration, which is described by this diagram right here. Glycolysis, Krebs electron transport chain. Oxygen is the final electron acceptor. And that's something that our cells do. Any aerobic organism, they do this. A lot of bacteria do this. And then we talked about anaerobic respiration that happens anaerobically. The main difference between what we just talked about in anaerobic respiration is that final electron acceptor. Remember, you can use nitrate, sulfate, carbonate, different kinds of things, and in the absence of oxygen. That is only done by some bacteria, some special bacteria. They aren't uncommon, but they're only certain ones do it. The other thing we wanted to remember is when we're doing anaerobic respiration, that the cells typically make a little bit less ATP than they would aerobically. So they would grow more slowly than an aerobic organism. The last part, we're going to talk about this little section that we've been ignoring, fermentation. And remember, fermentation is also an anaerobic process. Now, I want to remind you that human cells, muscle cells, actually can do some fermentation, but not all the time and not a lot. It can't maintain life, but it can help things continue for a little bit longer than they would without it. So right here, what you're going to see, so remember fermentation, it was under that part of our concept map that was anaerobic. And so what you can see here is we have glycolysis in fermentation. Glucose is converted into pyruvic acid, and we get NADH and ATP. But because we don't have oxygen, we can't do the electron transport chain, and it actually backs up the Krebs cycle too. And so we have to get another pathway to keep things going, and that is our fermentation pathway. And so here we have it tells you, we have fermentation end products. Well, over here is a more detailed example of what they're showing here. So here's fermentation. Here's glycolysis, our ATP or NADH, pyruvic acid, and some organisms will take that pyruvic acid and convert it to lactic acid. And that lactic acid would be the end product. Now look at that. That still has all these carbons hooked together. So there's still energy left in that molecule, but the cell's not able to get that energy. Lactic acid is what we do when our muscles are running out of oxygen. You go to the gym or you're doing, whatever you do to move around athletically, and your muscles start to burn. Why are they burning? Because you're generating lactic acid. You generate lactic acid in the absence of oxygen. What that's going to allow your cells to do is to generate a little bit of ATP, even though there's not very much oxygen around. And it can give you a little bit more endurance. It can let you work for a little bit longer than you would if you couldn't do that. Some bacteria do this. In fact, there's a lot of bacteria we use to make cheese, and these bacteria, a lot of them will make lactic acid, and the lactic acid is what curdles the milk and causes the lumpiness that we use to make cheese. Some bacteria, and actually some yeast, even some eukaryotes, will excuse me, got out of control here. There we go. Will use pyruvic acid in fermentation to make ethanol. And you're familiar with the idea of how beer and wine are made with yeast, and they use fermentation. So when you're making beer or wine, you have to take your fruit juice that has all the carbohydrates in it and seal it off, or whatever starting material you have, and seal it off so oxygen doesn't get in. Because if you get oxygen in, you'll do aerobic cellular respiration and you won't get ethanol. So you'll also notice that carbon dioxide is coming off, and that's when we talked about when you we didn't talk about it. I put it in the study guide that when you're making beer and wine, you actually have to have you can't just seal the container and let it happen. You have to have a vent for the gas to leave, and it's in the form of carbon dioxide. If you do not vent your fermenting liquids, the corks will blow out and it will explode. So we can make some products by fermentation in the absence of oxygen, and we just make a little bit of ATP. So it definitely, fermenting organisms do not grow anywhere near as fast as those that are using aerobic cellular respiration. This is just a diagram showing you a table showing you that there's all sorts of things that we make by fermentations. There's all sorts of companies that grow lots of microorganisms and produce different products. And here, I mean, we know there's lots of companies that make alcoholic drinks, but even things like acetone, formic acid, lactic acid. Butyric acid has a buttery flavor, and so that can be used in making different alcoholic drinks, and even there are fermented butters that you can use. Lactic acid is produced by fermentation to make things like sauerkraut and yogurt, cheese, and there's all different things. Different organisms. Oh, here's acetic acid. That's vinegar. Um, and and some of the acids, some of the fermentations produce a lot of CO2, which produces the bubbles in the Swiss cheese. So there's all different kinds of fermentations. It depends on which organism you use, because not all of them are able to ferment, and not all of them have the same enzymes to give you the same end products. The last thing I'm going to mention is just kind of a summary of aerobic respiration, anaerobic respiration, and fermentation, where I wanted to compare the maximum amount of ATP. If you put in 1 molecule of glucose and it's metabolized through aerobic cellular respiration, through glycolysis, Krebs, electron transport, oxygen is the final electron acceptor, you're going to get 38 ATPs. And those ATPs are going to be used to allow cells to grow. Anaerobic respiration, where you have different final electron acceptors other than oxygen, it just depends on the organism. You can get from 1 to 32 molecules of ATP from 1 molecule of glucose. And finally, fermentation, Candida albicans is yeast. There's no final electron acceptor, and you only get 2 molecules of ATP. So you're going to grow a lot more slowly with fermentation or any anaerobic process than you will with an aerobic process. And on that note, I'm going to say the end of metabolism.